All right, thank you very much for ARM for having us today. I'm looking forward to presenting, and I'll be making some forward-looking statements as we are a public company. Okay, so Fibrocell is a gene therapy company that is focused on the platform using autologous fibroblasts, and we use them to deliver genes of interest to the skin and other connective tissues in the hope to help rare diseases of those areas. And we are focusing on a few different indications. Our lead indication is in an autosomal recessive disorder called recessive dystrophic epidermolysis bullosa, or RDEB. It's a rare and just terrible skin blistering disease that I'll talk about a little bit more along in the presentation. And just for an update on this program, we're in a phase 1/2 clinical trial here. We dosed our first patient in February. We just recently had our DSMB review some of the safety data from that first patient and allow us to move into more additional adult patients. And we expect to report clinical data on this in the third quarter. We have a few designations from this with FDA already. Obviously, we have orphan drug status. We also have the pediatric rare disease designation, which is related in line for the voucher program upon market approval. And most recently, at the beginning of this year, we received fast track designation. We also have an indication for use called FCX013 in linear scleroderma. I'll only briefly talk on this today. This is a preclinical program we have that we're currently working on some non-GLP pharmacology and toxicology work in mice right now, and we're expecting to file an IND at the end of this year on that one. And for that, we also have orphan drug designation for that. And we also have work in arthritis and other related conditions, a little bit larger in scale, but is in early research phase. So just a quick glance at our programs, and all of these programs we perform in collaboration with Intrexon, which is a synthetic biology company that we've worked on for years on these programs and now moving into the clinic. So just briefly on the platform itself. So, the delivery vehicle we are using here is the autologous fibroblast taken from the dermal layer of skin. And, we use these to shuttle genetic constructs for areas for disease treatment. To do that, we're using lentivirus. We have a third-gen lentivirus that we developed. And we chose that one because of packaging size. A lot of our genes that we're looking at are quite large. And also the integrative nature of the vector helps with long-term expression in these sort of diseases that move on progressively through life. And these are all ex vivo modified cells. So we're talking about a gene-modified cell therapy program. And, we're looking to redose these patients. In a lot of these cases, they may use them for the rest of their lives the way that their disorder occurs. So just briefly on how the cells are manufactured from a high level, the cells are taken from the patient directly. We use biopsy punches to collect tissue. They're then expanded in the lab into small populations, which we introduce the viral vector and perform transduction work. And then once that occurs, because of the integrative nature of the vector, we then expand those populations to larger and larger populations, and then those cells are cryopreserved and banked in order for the patient to be able to use them, when they're ready in time, to have them thawed, washed, and then delivered back as dose to the patient the next day. We have a couple spots of cryopreservation into this process that gives us the ability to reduce the need to go back to these patients for skin every time they're treated, because we do expect to treat these patients, probably on some interval that we'll learn in the clinical trial for the rest of their lives. So this allows us to bank the cells with them for future use. So just a quick overview of our pipeline. Obviously right now in human clinical trials on our RDEB program and preclinical, as I mentioned earlier, on our scleroderma and research on 013. So I'll segue this into getting in a little more detail about our lead program in RDEB. So RDEB is caused by a single gene mutation. The gene is called COL7A1, and it encodes one of the collagen proteins, type 7 collagen. Now, the reason that's important is type 7 collagen, and I'll have a graphic on this on the next slide, it primarily makes up what's called an anchoring fibril, which is a mechanic sort of loop that goes from the epidermal layer to the dermal layer and then hooks around interstitial collagen, and then sort of that tight bond keeps your skin together. Think of it like Velcro, the hoop of the Velcro, and then the string below really is like the collagen fibers in your dermal layer, and they stick together and keep the skin together. So what unfortunately happened in these children, which are primarily pediatric, population is the lack of that fibril causes the layer of skin to lay on top of one another, and even just the slightest lateral friction can cause blistering. They then fill with fluid, and then pressure on that blister then spreads laterally. And you can see where now you get into this situation of constant wounding, rehealing, and then wounding again, and it causes all sorts of terrible issues, which I'll talk about on the next slide. So our construct encodes this COL7A1, a very large gene, but what we build in there is a synthetic version of the gene that we've done a lot of work on for characterization. And the goal is to locally deliver this to patients in the area needed, right in the area of wounding or in the area around the skin. And we plan to do this geographically around the whole body over the course of their treatment. So from the disease standpoint, when we think about what kind of clinical thresholds we need to target, this obviously we'll have to learn that in the clinic, but it's been very clear that the patients, when they're carriers, the adults, the parents of these kids, obviously are producing about half of what their normal collagen 7 is, but they don't manifest that clinically. So we already know that in order to sort of treat this disease, we think of it like almost like a spot welding effect. We really just need to figure out enough collagen 7 expression in the skin to form fibrils to keep the skin intact. And so we know we don't need to produce full restoration of the condition, but that we need to target, obviously we want to target as much as we possibly can, but the clinical threshold is lower than that. So this is a pretty simple drawing about, sort of how the disease works. If you look in these green anchor fibrils that are coming down from the epidermal layer into the blue layer, which is called the dermal-epidermal junction, they're present in our healthy skin, but in the children who have the disease, they're either malformed or they're null completely and they're not formed at all. And then they get that bilayer condition. It's progressive. It's obviously diagnosed usually right at birth because either the birthing process or shortly thereafter we're handling the infant tends to tear the skin. And they do have a high mortality rate. They tend to have a mortality from squamous cell carcinoma is primary, then sepsis, which is probably fairly obvious, is another problem. And about 76 of RDEB sufferers don't live into their 30s. So they do survive with this disease for a while, and then there's high mortality then. The current treatments, there are no FDA-approved therapies for this at the moment. Most of the care is palliative. It's wound care, bandaging, anti-sepsis bath using Clorox and other conditions to keep the sepsis layer down. There is a high output for these families. There's low reimbursement for their materials. They can spend as much as $10,000 a month just for bandages alone and antiseptics. And a lot of surgeries involved. There's a lot of finger separation surgeries and feeding tubes, and esophageal dilatations are very common. This is all from the scarring and sort of the reoccurring of wounding on these patients. The epidemiologist, it's ultra rare. In the recessive form, about 1,100 to 2,500 patients in the US. So just quickly, just through a high level on some of the data that got us into the IND. So, in vitro, we saw these modified cells. There's bottom panel there on the left are normal human dermal fibroblasts. You see some nominal amount of, the red color is the antibody to collagen 7 in human that you can see expressing from the cells. These are in well plates. RDEB, you don't see any expression. The same RDEB cells have been transduced above, and we actually saw hyperexpression of the protein in our replicates for the FCX007 product, which was very positive for us because, that's really the goal here is to produce as much collagen 7 as you possibly can. So this caused us to move into an animal model, and I'm sure as many of you are aware, in rare disease, there sometimes aren't models to really test these diseases. So working with Stanford University, we developed a xenograft model on skid mice. So essentially, human RDEB patients gave us skin for development purposes. We grew up their fibroblasts and keratinocytes and well plates and actually could create small xenografts that we then sutured onto the back of mice, and then that gave us an injection location to perform some pharmacology work on them, and that's what you see here with RDEB patients. Cross sections in the bottom. On the left is what our normal skin would look like. You would see the epidermis above that line and below. The green you see is the collagen 7 formation, so where you expect it in the space between the layer of skin. And after 17 days of injection of collagen 7, we actually saw nice expression in the dermal layer and the reseeding beginning of that white arrow of a new basement membrane zone. So this is the basis of what got us into the IND and gave us the ability to start doing clinical work. So a little bit about our ongoing trial. It is a phase 1/2 trial. We're enrolling 12 patients in total, 6 in phase 1, which are adult. So there aren't a lot of adults actually, but luckily Stanford keeps is part of a consortium of a large registry of patients that allowed us to do enrollment on this phase 1. The phase 2, we're having an additional 6 patients where we want to include pediatrics. But FDA asked us to provide some data before we got there. Some of the data we're collecting in primary endpoint is the safety of the product, obviously. And then there are some mechanisms we're looking at. One is, electron microscopy and immunofluorescence, can give us a feel about the actual structure of the anchoring fibrils. You can visualize them under EM. And then also the IF, what I showed in the previous panel is also what we're doing. We're collecting biopsies from the injected area on the patients. And we want to show collagen 7 expression by contact with the anti-collagen 7 molecule. And then those will be our mechanism endpoints, and we're also using 3D imaging and looking at wound healing. So the beauty of some of the modern technologies, we can actually do area analysis of these wounds and then look at not only wound healing rates, but durability. And we're comparing that to a matched untreated wound in phase 1 or a saline treated wound in phase 2 to give us at least some intra-patient comparisons of how our product is performing compared to another wound of similar area. So where we are right now, we're trying to walk our path through the trial. We dosed our first patient in February. Not uncommon in gene therapy trials, but after the first patient was dosed, we have a pause where we basically wait on the first patient, and a DSMB then reviews those data for safety. So that was 4 weeks post-administration, and we were happy to see that there were no product-related AEs. We didn't see any problems with RCL or with any autoantibody response as well. So that ruling allows us now, and what we're doing is coming up in the next few weeks, dosing additional adults in that trial. And then what the FDA has asked us to do is once we collected data and the adult population, which includes safety and benefit, that we would resubmit that, and then they would review that and then allow us to, upon positive review, begin to enroll the pediatrics, and we'll consider that in the phase 2 portion. So we're targeting the third quarter to report on these data. And, these will be the endpoints that we'll be talking about specifically. Okay. So moving on into our other program, I just have a couple brief slides about that. 013 is our product profile. We're using autologous fibroblasts encoded with MMP1. So if you're not familiar with the metalloprotease families, these are proteins, we're kind of doing the opposite thing. We're looking to break down excessive collagen with this target. And this one, because of the fact that we're using a protein that does that, we are using Entrexon's RheoSwitch technology. Now what this is an inducible promoter system where if you take an oral ligand compound, it then has a construct above the promoter that allows us to turn on and off the expression of this protein based on a lot of the dosing work we did in the animal studies that we're doing now. We felt we needed this extra layer of safety in this particular product. Because our goal here will be, and I'll talk about the disease in a moment, is to treat these really highly collagen, kind of excessive collagen areas that have been affected by scleroderma. And we're doing the localized version of that. You're probably very familiar with systemic scleroderma, and I'll talk about the localized version of that in a moment. So we're in the middle of a proof of concept animal study. We actually completed the in-life portion. It's a non-GLP pharmacology toxicology study just to learn more about what the dose should be, get some feel about the toxicology. And then what the FDA has asked us to do from there is then take those data and submit them with a pivotal GLP study, toxicology, and then that'll be our last step before we file an IND, which we have targeted at the end of the year. We've also been working on the CMC. Obviously, the manufacturing processes are very similar to our 013 or 07 program, so we're working on scaling that up now along with vector manufacturing. And as I mentioned before, we have received orphan designation on this, and we'll obviously be exploring other avenues as well. So just a little on the disease. If you're probably familiar with the systemic version, there is a localized version of scleroderma as well. It's usually diagnosed with an excessive production of collagen 1 and 3 that occurs in these sort of plaques or in these linear scars that happen on the skin. And they become thick on the epidermal and dermal layers, but also can be deep as well, can go in the muscle and even deeper. The problem with these diseases is, although may sound cosmetic, they actually can, especially the linear version, can go over joints and cause mobility issues, can cause developmental issues if they're not addressed early in childhood. The linear version tends to occur in children usually between the age of 5-15, so right in the developmental period. If they occur on the forehead, they can cause neurological disorders. And the current treatments are, really corticosteroids. There's been some work done with light therapy, and obviously just physical therapy trying to stretch the skin. So we think there's a real gap here missing that could be addressed with a gene therapy approach to sort of break up this collagen. And you can see why we want that additional control. You want that ability to shut that down so you don't continually break up tissue to the point of losing stability. There's about 160,000 people who have localized scleroderma of the linear type that are affected by joints. It's about 40,000, so a little larger population than before, but still a pediatric disorder. I'm sorry, an orphan disorder. So, just finally on milestones. So, we obviously completed some major ones for this year, which is dosing our first patient and moving through our safety review. We're looking to have our first human clinical data reported in third quarter, so we're coming up on a major milestone for our company, and obviously an IND filing on our second program at the end of the year. And there are some details on our financials here. We recently went through a public offering with some of our existing investors that put another $8 million on our book. That gives us enough cash to go into the second quarter. So we have cash through our data time point with some burn rate after that. Okay. All right, well, thank you very much. I appreciate your time.